So if you inhibit the paligenosis process in the pancreas with rapamycin, then the pancreas shrinks as it tries to repair and then just gets stuck there. And if you keep doing that, then the pancreas basically turns to, I don't know, the students call it snot. It just there's nothing much left of it. So you need that mTOR, and rapamycin inhibits that licensing step.
So hello and welcome to the Sheekey Science Show, where in this video I had the pleasure of speaking with Jason Mills, Herman Brown Endowed Professor at Baylor College of Medicine, where his lab studies this groundbreaking research on paligenosis, a fascinating cellular process that allows differentiated cells to revert to a more primitive state, enabling them to re-enter the cell cycle and promote tissue regeneration. Here we discuss all about this and more. Hope you enjoy. I wanted to reach out to you because I feel like we have similar research interests and the work that you're doing is very exciting. But probably before we get to some of the more technical questions that I have, we should probably try and get our audience onto a similar sort of page of understanding. So assuming that I'm not a sort of biologist and that we've only just met, how would you go about describing the different types of cells that you might find within the body and concepts such as differentiation or stem cells, cancer cells? How do you go about thinking about these concepts?
Oh my, that's a lot to put in lay people terms. So I guess we'll start off with that. Almost all the cells, if you're an adult, that you have are doing something, and they all have a task. And to accomplish that task, they specialize. So pretty much all cells are specializing in one type or a few related types of functions. And I don't know, like maybe one of the most common ones that people might know about are the cells that secrete insulin, because if you have problems with insulin secretion, then that can give you diabetes. So those cells are long-lived cells, and their job is to secrete insulin, and that's what they do. So that's a differentiated cell. And that's just one of the maybe 10,000 or so different types of differentiated cells that we have. And don't quote me on that, I just thought of that number. So even within, say, the pancreas, cells that secrete hormones, that one cell specializes in insulin, one cell specializes in glucagon, and so on. So throughout the body we have these specialized cells, and of course it makes sense that for maximal efficiency that almost all of the cells in the body are specialized, or what we call differentiated. And the reason we use that term is because at some point all of us came from just one cell, and throughout development there are both choices to either grow and expand, which is what you do mostly when you're growing as an embryo, and then to specialize along different routes. So those are called differentiation choices, because each time that a cell divides, it has the chance to differentiate and become sort of a certain category of cell, until we get to adults where almost all of our cells are, as I said, super specialized or differentiated. So those are differentiated cells. They're the mature functional cells in the body.
Cool. Yeah, no, I think that's a very good description. And so other people might also be aware about the differences between different tissues that we have within the body. So things like our liver has regenerative potential. So in terms of tissue turnover, how do different tissues differ in their ability to sort of regenerate?
Yeah, that's of course a huge question. The liver is interesting. It has regenerative potential. In fact, at least in experimental models, say with the experimental animals, if you remove part of the liver, the animals can grow back to a completely normal sized liver. So there's a lot of regeneration potential. The way my group specifically thinks about this question, because this is really what our interest is in sort of regeneration, as you brought up, versus how that regeneration can go awry, and cells keep thinking they're regenerating, but they're actually cancerous, and they're not regenerating, but they just keep growing and growing, versus not being able to regenerate, which we think is sort of one way to look at aging. So we're very interested in how that changes from organ to organ. And an organ like the liver is pretty much filled with differentiated cells. Pretty much every cell in the liver in an adult is normally doing its function and is specialized for that function. But when there's an injury, then some or many of those cells can be called back to a more progenitor-like, stem-like state, and that's how they regenerate. So that's how the liver does it, and each organ's sort of got a variation of how able it is to regenerate and how it does it. And some have mixes. So liver, we think now predominantly regenerates from the cells that are already there and are differentiated and are doing their job, and then they have to stop doing that day job and take a turn proliferating, dividing, and trying to repair the injury. Other adult tissues, and these are actually overall probably rarer, are the ones that have so-called professional stem cells. And those are cells that remain undifferentiated, almost like the embryonic state, like they haven't really chosen what type of work to do, and they stay in that kind of juvenile or sort of precursor state where they haven't decided yet which path they're going to take or which function they're going to take, and they stay in that state throughout life, so that very rapidly, if you have an injury or even the day-to-day wear, those cells can fuel the replacement of the differentiated cells. So those are, and then there's some organs that really are very limited in regeneration. Unfortunately, those are ones like the brain and the heart, and maybe there's a reason why those are the ones that wind up killing us in old age, because they're kind of the least able to regenerate. So those are kind of the three extremes. I'd say there's organs that come with professional stem cells that they keep around all the time to be able to regenerate. There are those that are pretty good at what we say facultatively, like they can, when the injury is bad, they can switch off their day job and repair. And then there's those that have very limited repair capacity. I think both the things that, like the heart and brain, are examples of that. A lot of neural tissue, although there's probably more than we used to think repair capability, but not nearly as much as liver or pancreas, or let alone the organs with stem cells.
Yeah, no, it's interesting because at least from school and perhaps even some of my undergraduate studies, we were really enforced that it was the stem cells that were doing a lot of the regenerative repair within tissues. Whereas, as your work has also established, the differentiated cells also play an important role in this in terms of regenerating and maintaining tissues. And so this kind of concept of going from a differentiated state to having some sort of ability to re-enter the cell cycle, I think some people come across this term cellular reprogramming. So the sort of going from a differentiated cell to a stem cell, which was the sort of famous Yamanaka research paper, which obviously a lot of people in the aging space are quite excited about. But whilst that seems like obviously it's a great discovery, it seems like this already is an intrinsic, perhaps, mechanism that cells have, this ability to go from a differentiated state and sort of partially rejuvenate. Now, how does the cell go about doing this? And what are some of the challenges that it faces?
Yeah, you gave me goosebumps because I'm so excited about this, especially the history, because I just find it fascinating. So I don't know. I'm not sure. Sometimes I bore even my own lab group.
I don't know. Please. I want to hear it. Yeah.
But you set me up for it. So yeah,
Thanks.
I think it's true that, say, before the Yamanaka explosion, right? And Yamanaka being, as you pointed out, the lead scientist who figured out that we could take, say, fibroblasts, which are differentiated cells from an adult, and run them all the way back to embryonic, like one cell, almost like state. And so then people are like, wow, is this something we can only do in the test tube? And then everybody started looking and realizing, oh, actually, like, there's a reason that cells could do this. It's because they can do it in our own bodies, and they're doing it all the time. So I think that's the way the field mostly. Of course, science is a great tapestry and landscape, and there are little pockets of people who are doing their own thing and maybe never believed that it was all a stem cell sort of thing, and the Yamanaka thing wasn't a surprise. But in general, that's how it happened in the field. Suddenly, everybody discovered, hey, it's not just a test tube function. It's something that cells do in the body. But what's interesting is, and I was one of those because I started as a stem cell researcher. And in some ways I was frustrated. In fact, it started for a brief time when I was working with those insulin secreting cells early on when I first started my own lab, and I kept trying to find the stem cells or the cells that were giving rise to them, and I couldn't. It just looked as though it was the adult cells that were replicating. And in fact, Doug Melton, of course, he's a big leader and has done amazing things in the field. Somewhere around the time that I was doing this, had a pretty important paper showing that, in fact, the way that you get more insulin secreting cells is that you coax the insulin secreting cells that are already there into proliferating, and there isn't a stem cell involved. So a lot of things like that started making people look more towards what we started talking about, the differentiated cells or the mature cells, or the idea of reprogramming that can happen in vivo. So that all sounds like it was all just invented with Yamanaka. So, and I forget how this happened, but I do like history, and now with all the Google books and the Hati project and these digital archives, you can dig back into the literature. And I stumbled across this amazing essay that was written in 1900 by the pathologist George Adami. So this is 125 years ago.
Wow.
and he was talking about the question of how do tissues regenerate, and
Nice.
and also how that might pertain to cancer. And basically everything that we just talked about is in that essay. So he says that there was this paper, and there was also a textbook that he wrote of pathology where he went into a little bit more detail a few years later. But basically, his idea was that tissue could regenerate either from mother cells, which was kind of what they called stem cells at the time, meaning cells that stayed capable of producing other cells, or maybe there were some rests, they called them. I mean, that means remains of embryonic cells that might still act like the embryo, and regenerate things. And the third way was clearly, he believed, and many people believed at the time, that differentiated or mature cells could also go back to a stem cell state and serve as stem cells. And not only did he believe that, but he outlined how that program might work, which is that when they're being differentiated and serving a function, then they must use all their energy to do that function, say secreting insulin, or in the case of some of my favorite cells, the ones that secrete digestive enzymes into our stomach and intestines. And the energy is going completely to that secretion of protein, and then if there's an injury and the cell decides it's got to be a stem cell, it's got to shut that energy off and retool itself, and then redirect the energy into dividing. And that pretty much sums up what everybody's been rediscovering in the last 10 or 15 years since Yamanaka. So I guess what I'm saying is that the concept has always been there. We got kind of overwhelmed with what I call the stem cell hypothesis, which is that basically stem cells are responsible for everything, and every tissue must have a stem cell because stem cells are the only cells that divide. And I'd say there's still a lot of debate whether there are actual stem cells in the liver, but clearly if there are, they stay really well hidden, and they're very few in number. And the vast majority of the liver, when you cause an injury to it that's big, gets regenerated by the cells that are existing there. And so really, I think as we're discovering the nice rediscovering, I should say, all this reprogramming ability, that it gives us a lot more tools for understanding both how cancers form and how aging happens. Because if you really admit that some 99.99, probably more, even another 9% of the cells in our body at any one time are differentiated and doing functions, and it's those cells that are the source for regeneration, then that's where you have to look for for most cancer, the cells that give rise to cancers in the adult, and also the cells whose regenerative capacity might decline as we age. So I think it really opens up a lot of doors to think about things. I mean, these ways that we conceptualize things are very important. If you're searching all the time for undifferentiated stem cells that are doing everything in organs where they don't really exist, then you waste a lot of time, because you're not studying what's happening before your eyes, which is this incredible ability of cells to actually reprogram, just the way Yamanaka made them do in the tissue culture, but they can do in us every day, all the time.
Yeah, absolutely. And I think this nicely segues into your lab's own research and this concept that you've now termed paligenosis. I'm very curious to know about why this research started and how you sort of came across mapping out this pathway.
Oh, yeah. Again, I think of course, I'm really excited about it. So the term is easy. Pali is the same. It's the Greek term for again or return. We don't use it as much, but everybody knows in palindromic sequences, everybody knows about DNA, that those are sequences that can go backwards and forwards. So that's the root concept of paligenosis, that cells can actually go backwards and forwards. And then gen is just, everybody knows generative, regeneration. That's the genus root. And then osis is a cellular process. So it's basically the process of being able to go round and around for cells to not only differentiate, but then to switch fates. That can be a de-differentiation, meaning go back to a stem cell state or going to some other state where they can serve as a progenitor. That it just emphasizes the plasticity. But how it came about in specifically our group to give this, I mean, first of all, this is just a term to get people to be talking about what we're talking about. It just gives people a context. Once you start thinking about reprogramming, then which people are beginning to do, most people then just focus immediately on the diseases that must be involved. That reprogramming is a part, or this plasticity, as we call it. Plasticity means that the cells can change their fates. They can become stem cells again. This idea of reprogramming and plasticity, then if people are studying cancer, then they automatically are interested in how the cells become cancerous. If they're studying regeneration, they're automatically interested in sort of the endpoints, how you get to the regeneration process. But the idea of paligenosis is, well, wait a second, if all of our, or which actually is probably not true, not all, but a large number of our differentiated cells in the body had this capacity, they're doing their day jobs, as I call it, secreting protein or digestive enzymes. And if they have the capacity to come back into the cell cycle as stem cells, that's not the kind of thing that is going to be evolved a different way in every organ. It just doesn't make sense. It's got to be a fundamental cellular process. It's got to be an osis, like mitosis and apoptosis. It should be an osis, and there should be an evolutionarily imbued way that cells can do this that's independent of tissue. So those are a couple of threads of thought. One is that we can focus on the process as opposed to just the endpoint. Do they become a cancer? Do they become regenerative? But what is the actual process the cells are doing to do this? What is the cell biology? What's happening to the organelles inside? How does the cell do this? Focus on the process, and focus on the things that are in common that across cells and across species. So that's the concept. And as you stated earlier, I think it takes a long time for new concepts to get out there. But I'm hoping by promulgating the concept, by coining a term and having people react to it and talk about it, we can start across fields, start to study the process, because we need people from every field to look at how cells do it in different tissues and under different circumstances, and define what the common themes are, the way that we know that cells undergo mitosis in different contexts, but mostly the core machinery of mitosis is the same. And the same with apoptosis or other forms of programmed cell death that are available to cells. Those, nobody really thinks of them as something that a liver cell can only do. Everybody thinks of them as something that all cells can do. So the idea behind paligenosis is it's something that all or almost all cells are able to do. So what are the common elements of the process? How can we study it in a tissue-independent way.
Exactly.
So I think, part of the issue, of course, is just getting people to understand the term. And then the other part is technical. You can make cells undergo apoptosis or mitosis. In fact, the main thing that you do in tissue culture is, like, you wouldn't have tissue culture if you didn't have cells proliferating. And when they proliferate, they undergo mitosis. So almost the definition of tissue culture is to have them grow, which means, so you can study mitosis. And that's why we've been studying mitosis forever, basically, as long as we've been studying biology, because we can observe it. And apoptosis, the programmed cell death concept, came later, late 60s, early 70s. The term was coined in 1972. But we can do that also in vitro. But it's very hard to study the exact cellular process that occurs when cells are in tissue and are differentiated, and then within the tissue they get a series of signals that make them reprogram in the tissue, because as soon as you put them in culture, then they're growing all the time. They're not differentiated, long-lived cells the way they are in our body. So I think that's what sort of held us back from even thinking of this as a concept. It has to be done in basically in organisms and looked at, and so we don't have good, and even now it holds us back because we still haven't come up with a really good model that we can study it in tissue culture.
Yeah, no, absolutely. And I think having read your work, the one question that came to mind was how do you study it? And it seems like definitely in vivo is the better way to do it, but then that comes with additional challenges compared to in vitro studies. But I guess one of the questions that you just made me think of was, what is this trigger that the cells are sensing that initiates paligenosis? Like, what is it that they're sensing, and how are you trying to simulate in the lab?
Yeah, so that's an open an open question. We keep trying to get earlier in the systems that we study to try to find the common denominator, and the earliest signal that we can find is what's called reactive oxygen species release. That there's a burst of these reactive oxygen byproducts. They're ionized and that's what's going back to my chemistry, atomized oxygen species that are very damaging to the cells, but also serve in lower doses as cellular signals that start processes, normal processes even in the cell, but at very high doses can damage DNA irreparably and either cause cell death or increase the risk for mutation and potentially eventually cancer. So that the initial things that seem to happen in all the systems that we've studied is a large amount of reactive oxygen species. Now, upstream of that may be specific to the type of injury or even the tissue, but downstream of that, there seems to be a lot of similarity that the reactive oxygen species response molecules in the cell are very important. The way that the cell then reacts stereotypically is to begin to what we call enter the autodegradative phase, which is basically the first phase of paligenosis, and that is where there's a massive upregulation of the cell's degradatory recycling machinery, lysosomes, and autophagosomes, or the self-eating function or autophagy. So those are the earliest common triggers, is this increase in reactive oxygen species and a massive upregulation of lysosomes and autophagosomes, which that of course makes a lot of sense. And it's because if you're a invariably large differentiated cell, you're larger because you're trying to perform your function, like making and storing a lot of things that you're going to secrete. If you're going to go back and become more like a stem cell, you don't need all that, because you're, as my analogy that I use several times, you're giving up your day job, so you don't need all of that equipment, and so that's invariably the first stage, it seems, and it seems to be triggered at least in part by a reactive oxygen species. But it's going to be complex, the triggers, and those will vary from tissue to tissue and condition to condition. Some mixture of inflammatory signals, responses to the destruction of tissue around the cells that are what we call non-cell autonomous, meaning outside forces that impinge, and those will probably have to be varied. But invariably, it seems there's this massive upregulation of recycling machinery, which is almost a no-brainer whenever we talk about it. Everybody says, oh yeah, that makes sense. But then when you see it, it's just amazing. Like half the cytoplasm of the cell becomes these autophagosome and lysosome structures. I mean, they really get at it. Trying to, and actually, I know that you mentioned about we have a paper that's under review now, touch wood, but is out there as a preprint where part of that phase is not, at least in some cells, we don't know how universal to the paligenosis process it is, but at least in some cells, in addition to all the internal recycling, the cells sort of seem to just give up and just jettison. They just shoot material out into the lumen because they don't need it anymore, or maybe it serves also other secondary benefits, like, for example, if you're under a bacterial infection, shooting a bunch of charged membranes out into the lumen could tie up the bacteria. So that's all the sort of first stage, massive recycling, and the triggers is oxygen radicals.
Interesting. Yeah, you just brought up so many points. So firstly, I agree that it seems like there probably are various different signals that the cells have to respond to, and it's going to depend on the cell type and the tissue as well, and the severity of that stress. And then coming back then to triggering paligenosis, one of the things that eventually happens is it re-enters the cell cycle. And I always, I read something very interesting earlier this year about 2 ways in which cells can rejuvenate. One is through cell division by diluting, essentially through cell division, it could rejuvenate or dilute the damage within the cell. And then the second mechanism is by secreting things, which, as you just sort of mentioned there, in that initial phase, could also be that secondary mechanism at play. And so the question I really just came to me was this paligenosis phase, how long does it take for the cell to go from a differentiated state back to this re-entry into the cell cycle?
Yeah, no, so of course that awaits a lot more analysis across tissues to see what the range is. But in our favorite system, which is really where we discovered, and I probably should make a little plug for it. I mean, the best system that we know of for studying this inside tissue is in the epithelium, the lining of the stomach. And the reason for that is because the lining of the stomach is exposed to all this acid that the stomach itself secretes, plus everything we eat and take in. So it's an incredibly harsh environment, and it's where the body first deals with whatever crap we might have eaten, especially historically, evolutionarily, how much stuff that we had to eat to survive. So the stomach is that and to kind of cope with that environment, it has a professional stem cell, as we were talking about earlier in our discussions, a small, undifferentiated, relatively rapidly turning over cell that constantly is dividing, and the reason why we probably have that in the stomach is exactly for all that harshness to feed this constant turnover of the cells at the surface. So there's a professional stem cell, but much further down, deep within the glands, are the cells that secrete digestive enzymes. And I'm getting to answer your question, but I just want to talk about how we know the answer, but also to give a plug for how the stomach shows right there with its own sort of internal control how there is the regular stem cell and now there's a separate paligenosis process. So those regular stem cells are dividing all the time, and if you do something to injure the stomach, it's very rapidly responsive because, as you might think about it, we've got all this stuff that we've eaten and a whole bag full of acid sitting, right in the middle of our abdominal peritoneal cavity. And so the stomach has evolved to be incredibly responsive to trying to shut down any sort of damage that might allow everything to leak out into our peritoneum, into our abdominal cavity, and kill us. So it's very responsive, and the stem cells can respond very rapidly and increase their proliferation. But the cells down at the bottom, the digestive enzyme secreting cells, can live for years, or at least in our mouse model, they live at 6 months, 9 months, a year without ever doing much of anything other than secreting digestive enzymes to digest stuff. It's those cells, though, that you can injure the stomach so in a way that the stem cells are not capable of, or sense that they're not capable of repairing by themselves, and you call these very large digestive enzyme secreting cells back into the cell cycle by paligenosis. Okay, so in some of our models, those large cells that are just wrapped with rough ER and ribosomes and mitochondria, all of that can be, well, the vast majority of it can be auto-degraded and cathartocytose jettisoned, so that the cells enter the cell cycle within maybe 40 hours or so, so less than 2 days. A sufficient stimulus can reprogram those cells to become regenerative cells again. I don't know if there's systems that are faster. My suspicion is there probably are, and it probably depends. The liver, for example, I think is probably faster at doing the coming back into the cell cycle, and it may just have to do with how much stuff the cells have to deal with to get rid of before they can then divide. There's also a check. I should make one other point about the concept. So the reason why we know there's stages is, and the first stage is this auto-degradation stage, is because we inhibit the cell's ability to recycle or degrade things. For example, using everybody's favorite drug coming out of the pandemic, hydroxychloroquine, which I don't know what it does in the case of COVID-19, not too much. But what it's very good at is reducing the activity of lysosomes. And those lysosomes are the part of that recycling machinery. And if you give cells hydroxychloroquine, then they can't undergo the auto-degradation, and the whole palingenosis sequence fails. The next step, which is what I was getting to, which is probably the most important or the second most important for the delay, for the kind of broader answer to your question about how long it takes. The next checkpoint is the most important one for cancer, and that is a p53 mediated checkpoint. And it's again, the first stage is kind of a no-brainer. The cells have to get rid of all that stuff to become smaller and to become stem cells again. And the second step, the second checkpoint, is also one, if you think about it, you're taking these long-lived cells that could be there for years and could have accumulated who knows what mutations and not really been bothered by them because all they do is secrete the same things over and over again. And if they have a mutation in some gene for proliferating, who cares? But now you're calling them back into the cell cycle. So the cells have evolved a second checkpoint, which is are you okay to come back into the cell cycle checkpoint? And that one is probably the most important in terms of rate limiting step, if we think of a chemical reaction and the delay, and it takes them a while to decide to do that. And it has to do with titrating the p53 levels down enough so that the cells are freed to go into the cell cycle. If the p53 levels stay high, then they're slower. If they stay really even higher, then they probably undergo a senescence-like aging-related sort of state. And if they're really high, then they die. So they're slowed by that. So putting that all together, if you could speed up the process if you speed up the checkpoints. And we haven't actually done this together, but you can go through the auto-degradation phase if you give a drug like rapamycin, which increases auto-degradative activity. And so that they'll go through faster, and they'll definitely go much faster into the last stage if you knock out p53. So cells don't have that checkpoint anymore. Now, of course, they're entering the cell cycle with all kinds of damage, and that increases the tumor risk, and that's why they don't do it normally. So I think actually it's a good question. So, kind of I said a lot of stuff, but just to kind of summarize how fast how quickly a cell can go back into the cell cycle is a function of sort of probably how differentiated it is. So how long it takes to get rid of the all its organelles and structure that it needs to do its specialized function. But more importantly, how long it takes it to clear the quality control step that says come back into the cell cycle. And you don't want them coming back too quickly because leaving around a lot of those damaged organelles can increase damage. And certainly coming back into the cell cycle when your genome and all your DNA maybe still be damaged and not repaired is a very bad idea for cancer. And so, in fact, that's why there's this probably, in this time sense, there's this balance between regeneration and cancer. The faster you go through, the more likely you are to make errors when you go through. But of course, the more slowly you are, the less able you are to quickly repair damage. So I think that's the, it's a deeper question than just how fast can it happen. I have a sense that if we could like ramp everything up and have a model in tissue culture, they could probably do it in about 12 hours or something. But inside of tissue, by the time they get the signal, by the time they decide, oh, this is a real deal, and launch into it, which is 6 to 8 hours in our model. By the time then they degrade everything, which is another 8 to 10 hours or 12 hours, then there's that kind of long checkpoint before they're allowed to go through.
Yeah, no, that thank you for clearing that up, because obviously the reason I think I first came across your work was because I myself study P53 and senescence and the decision making between apoptosis, senescence, and I guess now paligenosis.
Yeah.
And so before we come back to maybe more senescence and aging, just to sort of wrap up my own understanding of paligenosis, once that cell's now re-entered the cell cycle, how does the process eventually sort of end? Is it just the matter of the cell eventually then differentiates again? Or what is the end point?
Well, it definitely can differentiate again. Beyond that, we don't really know that much about the options next. I mean, clearly, we know that if it sort of fails to license properly and that you take cells with genomic instability that have oncogenes, mutations that drive tumors, then they can develop tumors out of it. So they never return. But I can say that in our systems where we give toxic doses of drugs to the stomach that specifically target the cells in the stomach, and we just do it for a couple of days, then the entire stomach basically reprograms. And the cells at the base that are capable of undergoing paligenosis in response to the drugs pretty much all undergo one round on average of proliferation, and then we invariably don't keep giving the drugs because they're toxic to the mice. And then the mice recover almost always, and they come back out of the cell cycle and they go right back to being digestive enzyme secreting chief cells. So one thing that you said about apoptosis versus mitosis and P53, one way to look at paligenosis is it's just the differentiated cell's way of licensing whether it will undergo apoptosis or mitosis. I mean, I think there's middle ground there, but the simple way and the way that we look at it when we're just starting to characterize this process, so that we're looking at more extreme cases of what we call failure of paligenosis, which invariably in our which not invariably, but in our conceptualizing, that means that the cells just die. So we're looking for big failures, and the big failures just cause death. So essentially they fail the licensing, so that's apoptosis. If they succeed in the licensing, then that's mitosis. So in some senses, paligenosis is the process of licensing apoptosis versus mitosis in differentiated cells. Now, there's going to be more complexity to that. There's going to be in-between outcomes. Some of the failure causes other types of death besides apoptosis. We already know that ferroptosis can happen and so on. But the simple way to think about it is that way. And the simple way that we think of P53 is that that's actually its most important job. I mean, we think the easiest way to look at P53 is that it evolved with multicellularity the same way paligenosis evolved with multicellularity. So going very back to the first, and we'll stick with animals, the first multicellular animals, the whole point was to specialize and to develop cells that have specific, differentiated, specialized functions. Like we started the whole conversation at the very beginning. That's why you become multicellular and just don't stay as a little protist swinging around by yourself. Is because you want to be able to specialize and have cells that do digestion and cells that do nervous and sensation nervous system functions and so on. But as soon as you start doing that, then you give up what the protists can do, which is whenever they feel like it, decide to divide in half and make another protist. So then, how do you reproduce in that multicellular state, and how do you repair damage if the whole point of developing multicellularity was to have everything specialized? And so our concept is, as soon as you do that, you have to develop or evolve a mechanism, a paligenosis, basically a mechanism for taking the same cells that became differentiated and running them back into the cell cycle. But the problem is, what if they have accumulated mutations in that time and they're genomically unstable, and then they overgrow and threaten the life of the host? Then you need a mechanism for protecting the host against that. And that's where p53 comes in. So obviously there's a lot more complexity than that, but the simple way to think about it is that paligenosis and p53 are inextricably linked, and they were evolved as part of multicellularity and licensing. And I tend to think that, like in our stomach model, where we have a professional stem cell and the paligenosis happening, and we can do one or the other in different scenarios, the only one that cares about p53 is the paligenosis cells. The actual professional stem cells don't care about p53, whether it's knocked out or they're not using p53. They don't express it. They're not using it as a check. It's only the differentiated cells that are using it.
Interesting. Well, firstly, before I forget, I think I knew from the Yamanaka research that p53 is one of the sort of proteins that inhibits or acts as a roadblock to going back to the stem cell. But I guess my other question was more about, how does the cell, given some sort of stress, distinguish or decide between paligenosis or entering something like senescence? Because on paper, the two pathways are similar but very different in that one is re-entering the cell cycle whilst the other is exiting the cell cycle. But they're both, at least senescence is still important for rejuvenating or regenerating tissues because of all of the things it secretes into the environment. So in paper, they both seem very beneficial for tissue repair. So how is it then determined what should happen?
Wow. Yeah. I mean, of course, we're, again, super excited about this, and I don't know too much right now, except I'll give a plug for a colleague of mine who also happens to be my wife and who is in the lab next door, who works on aging and senescence in tissues. And through our occasional, well, actually yearly lab retreats, we began to realize that as my lab's been working on this paligenosis concept and p53 licensing, and her lab's been working on tissue senescence and aging, that in many ways we're studying the same process, but in different contexts and with different outcomes. So I will say that we're quite sure of that. That in fact we've got a paper also in bioRxiv and in round 3 of re-revision or something like that with the 4 reviewers that should be coming out any day now from the 2 labs, where one of our absolute evolved with p53 regulating, although we don't talk about it in this paper, that's the next paper, paligenosis genes is also, when you knock it out, a great model in and of itself for aging and senescence in the bladder. So the ties, or I don't even know, sometimes I call it the 2 sides of the same coin, but as kind of you pointed out, it's not, it's almost like different shades of outcomes, but it's all the same system. And it's different levels of p53, different levels of these paligenosis checkpoints can lead to either a regeneration phenotype or a senescence phenotype or a death phenotype. Beyond that, I don't know. I mean, yes, senescence has some wound healing properties that are good, but obviously too much is a bad thing also. So, but the machinery seems to be similar.
For sure.
And I think in fact, a lot of the ways that the paligenosis concept has helped us and my wife's lab, and we're hoping, we actually tried to get a meeting together, which we were super thrilled about, and every one of the speakers immediately said that they were thrilled about it. And the meeting was about half sort of paligenosis regeneration type people and half senescence and aging type people. But so far we've been turned down by 2 or 3 different meeting organizing outlets, I guess, because they don't quite get how these two fields are related. But I guarantee that they're going to be related. And everybody that we invited immediately knew that they were, but we don't meet together at the same meetings. And if we did, then we could start answering those questions. I think it's going to be simpler than people think. You just have to stay in tissue for one thing. I think tissue culture models are misleading because they're almost always, first, they're even with organoids, which are very similar to the in vivo situation, which is what we study, but even with those, you're constantly driving proliferation. So you're never going to have a real palligenosis licensing step. And it's hard to know exactly what senescence means in cells that are tumorous already. So we need to be able to know what senescence and palligenosis and recruited mitosis and failure and along those means in tissue. And, that's tough, but we have to start doing it together.
Exactly, and I really hope that this does happen, because I think, as you said, the components, the biochemical components, p53, mTOR, are both important in the pathways. And speaking of mTOR, obviously rapamycin is known as being that one sort of longevity compound that continuously extends mouse lifespan in different variations. And so given that obviously mTOR activity is needed for the cell to re-enter the cell cycle and rapamycin is an inhibitor of mTOR, how does that influence our understanding of the benefits of paligenosis?
That's a great question. I mean, so we know about rapamycin and paligenosis in every setting, including sort of the OG, as we call it sometimes, Yamanaka paligenosis, which is sort of a paligenosis in vitro. I said we didn't have a model for it, but it's very hard to study there because if you know anything about it, it's like the number of cells that go through is very small, and there's a lot of stuff going on. But we know that if you treat with rapamycin in that system or in any of the systems that either we've directly studied or we sort of interpolate that we see other people are looking at regeneration. It looks very much from the data they presented, even if they haven't thought about it as paligenosis, that it is paligenosis. In all those cases, rapamycin is a deal breaker. It just doesn't happen with rapamycin. On the other hand, it does partially happen. So in the systems we know best, what happens if you treat with rapamycin is, as I said, that autodegradation phase goes super fast because rapamycin encourages autophagy and autodegradation. So they zip right through the getting smaller and jettisoning and recycling everything that they want to. In systems where they also rearrange their chromatin to be more juvenile, they do that fine with rapamycin, but then it completely halts. So they don't progress beyond that. They just can't re-enter the cell cycle. So if you injure the pancreas, which is like liver, one of these organs that needs paligenosis to be able to regenerate, I mean, it doesn't have stem cells, or at least if it does, they're minute and don't contribute very much. So if you inhibit the palligenosis process in the pancreas with rapamycin, then the pancreas shrinks as it tries to repair and then just gets stuck there. And if you keep doing that, then the pancreas basically turns to, I don't know, the students call it snot. It's just there's nothing much left of it. So you need that mTOR, and rapamycin inhibits that licensing step. Now, that said, even as you brought it up, which is why it's great to talk to people, especially across fields, the interesting thing is that a lot of those knockouts, like the one that mice with genes deleted that have catastrophic palligenosis fails, where the cells just die, don't undergo senescence and just die. If you treat with rapamycin, they don't die. So it's sort of upstream of that licensing, and what that like the one of those genes, the genes we know best where that happens, is the one that's also the one that has the premature aging phenotype in the bladder. So it's possible that regulating the levels of regeneration, maybe exuberance or something, through that gene and through palligenosis via rapamycin at lower levels maybe helps in some ways modulate the process. I think it's kind of paradoxical though, because I tend to think of palligenosis as rejuvenating, and if you're blocking rejuvenation, then that seems like aging, but You would also be blocking the death if your licensing was failing. And so you'd keep the tissue still around rather than having it die. And you might also block the p53 senescence. So in that sense, you may stop large repair. Like for example, maybe these mice on rapamycin, if you went in and injured their livers in a big way or their pancreases, they would do very poorly and much worse than other. But as long as you keep them in a cage eating food and not under any kind of, because in a sense you could go through your entire life like a mouse in a cage with constant food and water, and not need palligenosis at all. You just wouldn't need it because your normal stem cells are doing their things, and you just every once in a while have these other organs like liver and pancreas with at the very day-to-day level, they can just, the differentiated cells don't have to undergo palligenosis. They can just divide as differentiated cells, and that's how they can maintain the tissue. So maybe they don't need it. But I think that would be the issue. The question is, if you are in the real world where we do have injuries and we need palligenosis to regenerate, it may not be the best thing to be on rapamycin at that time. But if you don't need it, then it could stop a lot of senescence happening potentially that may be the failed outcome of palligenosis. I think that's a pretty good explanation.
No, I like it.
Thank you for asking that question. Yeah, I hadn't thought about it in connection with the two.
Yeah, and I think it's not just senescence. I think also perhaps if it's blocking the full palligenosis, it's also reducing the likelihood of cancer. And obviously,
yes,
a lot of mice die of cancer, so that's still another potential explanation.
It absolutely would, yeah.
And that kind of, to try and wrap up our conversation somewhat, the other thing that the aging space is obviously interested in right now is finding regenerative or rejuvenative therapies. And one of them, of course, is extrapolating from Yamanaka's discovery of cellular reprogramming, but not wanting to do that because in vivo, if you're going back to a stem cell, that cell's lost its identity and could become tumorigenic. And so instead, you may have come across the term of partial rejuvenation, which in many ways kind of just sounds like paligenosis. And so many people are now trying to find ways to sort of partially rejuvenate a cell so that it can regenerate without losing its cell identity. And from talking to you today, it seems like you're doing exactly the same thing. And so I'm curious to know if you've thought much about how best we could be finding therapies to slow down the aging process or to trigger rejuvenation.
Yeah, no, I mean, I think I've kind of said everything I know about it, but except for one interesting thing that kind of relates to what you were just talking about. So actually, I don't know about the partial rejuvenation, but an area where palligenosis seems to be bad is in the brain again, and in neurons specifically. I'll take that back. I think actually glia are really good at palligenosis, and that's in fact what they do. Mostly they almost spend all their time differentiated, and when there's a need for a lot of glial cells, then they clearly seem to reprogram. And we haven't studied it, but from piecing together the role for mTORC and p53 and autophagy and all, it looks as though they're doing the same sort of thing. So, but neurons can't really come back into the cell cycle, but they can undergo sort of a partial rejuvenation, at least fetal, embryonic, or early juvenile dorsal root ganglia. We know that they use exactly the same sort of 3 stages. They don't come back into the cell cycle, but they use the same sort of autodegradative reprogramming chromatin, and then they're able to extend neurites again. So neurons can kind of do that early on, but as they probably age, they can't do any of it particularly well, and it's probably a bad idea for them to try to do that. And in fact, that's been sort of an emerging and not entirely unexpected, but kind of interesting that in terms of fields I didn't necessarily expect would occasionally take up the palligenosis term. It's been in some of the neurodegenerative spaces where they've been trying to explain why neurons look as though they're almost trying to come back into the cell cycle, upregulating CDKs and such, and that definitely seems to be a bad outcome. And so it's possible that in that space, specifically neuronal space, that palligenosis is bad because the cells are trying to reprogram an injury, and then it just leads to p53 or haywire somehow. But how to really target? I don't know. I put that a lot of times, like when I'm trying to get meetings together, like what I was talking about. But I guess we learn certainly that we have drugs that can target it, and rapamycin was a great example. So I just think that I think that when people are studying things like partial rejuvenation, if they came at it from a palligenosis context, it might be helpful in the sense that what they're studying is a conserved series of cellular events that are independent of the cell type, and those events, if you thought of it that way and thought of the checkpoints and thought of the specific genes involved, maybe you could get at exactly how to modulate or titrate or figure out more what's going wrong from a more general cell biological point of view rather than whatever the specific tissue point of view that people tend to approach those questions in. I think aging, I don't know as much, but I've been getting into it more. Any day now, we're hoping to get our first notice of award on an aging award with my wife, who's the contact PI, principal investigator on that. So I'm learning more, but I think my sense is that there is some general concepts of aging that go across tissues, but we're still quite siloed in how we approach the overall process, and especially when we talk about it as a generalized sort of cell biological process. The exception to that being the senescence field, but then I think often that's done in the cell culture context, and so it's almost too generalized and maybe on the model that's not the most directly translatable into tissue. But then when we get into the tissue, it of course is harder, then people tend to be focused on their specific tissues. So I guess maybe that would be my advice, at least, is to try to find common aspects across tissues that because any drug that we're going to want to use to target is going to affect all the tissues at once. So we probably want to understand the overall process. But I need to know more too about what people are talking about with partial rejuvenation, and does it sound like a failed palligenosis, or does it sound like regular palligenosis?
It sounds like what they're searching for is paligenosis. They just don't realize it already exists.
Right. Yeah.
That's my theory. But I'm also relatively new to this concept. So,
No. Yeah. Right. I mean, we've been dealing with it for I guess 6 or 7 years, or 7 years maybe in the lab, and so it comes up all the time. Is this palligenosis or isn't this? And to me, what palligenosis, we defined it as those stages, but what it really is getting people to talk about the shared cell biological issues of how you license and how you choose, in response to injury, senescence versus mitosis versus death. So, and in a tissue independent way, how cells do that. So.
Exactly, and I'm really hoping that this discussion today, and people listening, that will be a sort of trigger for more people to learn about it and to think about it. And so just to sort of, I guess, summarize and to thank you for coming on today, where can I direct my viewers if they want to learn more about this pathway or to help contribute to this field?
That's a great question. I mean, we have reviews out there pretty technical. I think our favorite one we spent about 5 or 6 years on with 2 co-authors who both now have their own labs studying paligenosis. One is going to start his lab at Emory and one at WashU, and that was in the IMBO journal that just came out a couple of months ago. It's got a lot of kind of general stuff in it, so I think that's a pretty good review across anybody who's reasonably biology savvy. There was, when we first had the term, there was a nice Popular Scientific Press piece that came out in, I think it was in Quanta magazine. So you actually, if you Google for paligenosis, you can find it eventually. And that was actually in the first, and I think it still holds pretty well. So those are two routes. And if you have specific questions, I'm happy to take emails. It's expanding into areas one pocket at a time as people start to think, hey, maybe this is going on in my tissue too. And there's been some very interesting, people that have reached out to me, not just in the aging, but in the atherosclerosis field, for example, as you're arteries harden, there's all kinds of reprogramming of cells, and it looks like that might be by a Paligenosis mechanism, which means it might be targeted by our drugs and so on. So people from different fields have reached out already. Happy to entertain such requests directly.
Nice. I'll put some of those links in the description. So yeah, thank you again for coming on today. It's been great talking to you, and best of luck with the continuing research.
Oh, thank you. Yeah, no, that was fantastic questions, and I was super excited. And the way you rephrased everything, as I said, was kind of hit directly how we came about it and how we're thinking about it, and we're excited about it.